So, good morning. Today we will discuss about the instruments which are used in the microbiology lab, and the main instrument which is used in the microbiology lab is a microscope. So first, if we go into the definition of a microscope or microscopy, so microscopes actually are the instruments which are designed to produce magnified visual or photographic images of small objects. So that means microscope is made up of two words, micro plus scopy. Micro means small and scope means to see. So that means these are those instruments which are used to see the micro things. So we can see very the things like bacteria, viruses, and there are different kind of microscopes. So what microscope actually do? So microscope magnifies the image and separate the details in the image. If you magnify anything, the details becomes separated, and and finally those details can be visible to our eye, which were not initially visible. So there are many types of microscopes nowadays present. So there's simple microscope, compound, phase contrast, dark, dark background actually, and this is a fluorescent electron microscope and many others microscopes. But basically we can divide the microscope into mainly two different types on the number of basis on the number of lenses present in the microscope. So one is a simple microscope and another one is a compound microscope. So simple microscope means a single lens. If we are using a single lens, or that is a magnifying glass, so that is a simple microscope. But if we use those type of magnifying glass in a combination and more than one lens we are using, those microscopes are known as compound microscopes. And then on the source of illumination, that is a source of light, in those they are further divided into light microscope and electron microscopes. So in electrons the source is electrons, where in light there is light microscope. So today we will a little about the compound light microscope. And then the light microscope is further divided into different types, like bright field, that is a dark field, phase contrast, and fluorescent microscope. And one is laser confocal microscope. And so simple microscope, as we had already discussed, this is a light, only one lens we are using, that is a magnifying glass, pass through the object, and the two or more lenses if we are using, that is a compound microscope. We see this is a picture of light microscopes, and it has many different parts. So broadly we can classify these parts in the lens system or an optical system and the mechanical system. So if we go for the mechanical systems, the mechanical parts actually hold is provide the structure or scaffold to the whole microscope, that is to the optical system or lens system. So it has, you can see, let's start from the upper side. So upper side you can see these two things and lenses. Actually these are the lenses, and these are known as eyepiece and ocular lenses. Then they are joined here, and this is usually known as a draw tube to the main body of the microscope. Then here you can see this is an arm. So in arm, it is to hold the microscope, to carry the microscopes, and there is a revolving nose piece. So that means it is a nose piece, a part of the microscope, mechanical part, and which holds on which there is a there is a nose piece which can be revolved, and that there are certain objective lenses. Usually 3 or 4 objective lenses are mounted on it, and we can revolve it and bring any of the objective lens into the path of the light. Then there is a stage to keep our specimens over here. Then there are sub-stage condensers. Then there is a which condense the light o- onto the object. Your object is kept on usually on the stage. Then aperture diaphragm controls, so it controls the light, how much light you want, and there is a light source here. Right? In some of the microscope there is a controller also, like intensity control, and some don't have. And it is the two knobs are present over here, and the one is force focusing. These are actually the adjustment knob, and so we use these knobs to focus our microscope on the object. And this is the fine focus knob to fine adjustment, force focus knob